Before we start to talk about virus replication, for example, influenza or HIV virus life cycles, it's important to be able to recap the structure of a virus particle. Now, we know viruses are non-living particles that must bind to and infect a host cell to be able to replicate inside the host cell using its machinery, such as its ribosomes. So a virion, you can pause the video possibly if you like to go at the quiz, and then we'll go through the answers very shortly. So number one, the virion then, well, that's the infectious virus particle. Now we know that can enter a host cell and it can replicate inside the host cell using, for example, the ribosomes for translation of the virus genes. Now the capsid, number two, is the protein coat surrounding the genetic material of the virus. So a capsid can be made up of different types of proteins that give the overall outside shape of the virus. The nucleic acid within the capsid, number three, that can be one of three types. It can be single-stranded DNA, it can be double-stranded DNA, for example in bacteriophage viruses that infect bacterial cells, or it can be single-stranded RNA. Now, viruses with single-stranded RNA are called retroviruses, and these include both influenza and HIV virus. So most viruses that infect animal cells and plant cells are retroviruses with single-stranded RNA inside. So number four, the retrovirus then, we know it has the single-stranded RNA. Influenza actually has 8 single-stranded RNA molecules inside the capsid, and these will then be used for transcription and translation inside the host cell. Now the enzyme RNA polymerase then, well, for example, influenza virus has this enzyme inside the capsid, so when the virus infects the host epithelial cell, the RNA polymerase will actually synthesize a new strand of single-stranded RNA against the template strand of RNA, and the new strand of RNA can then detach, and it can attach to a ribosome, and it gets translated directly into the viral protein. Now DNA polymerase obviously will allow the formation of a single strand of DNA from a single-stranded template DNA strand. Now, both RNA and DNA polymerase, they do not catalyze the formation of hydrogen bonds between complementary base pairs. That will just occur naturally. But the nucleotides that align next to the complementary exposed base, they're actually linked together by the activity of the polymerase enzyme. So RNA polymerase joins together RNA nucleotides. It forms the phosphodiester bond that links the nucleotides together in a longer polynucleotide chain. DNA polymerase links together DNA nucleotides, again by forming the phosphodiester bond, and that will form the sugar phosphate backbone of the brand new strand of DNA. Now, an antigen is a molecule on the surface of a pathogen, whether it be a virus or a bacterium, against which the immune system of a human will raise specific and complementary antibodies. Now we know antibodies are quaternary proteins that have a specific shape in their variable portions, and they will bind a specific antigen on the surface of the pathogen. So we know virus particles outside the host cell are inert, and that means they cannot replicate on their own. So they must get into the host cell to be able to replicate. Now, viruses can have one of two types of life cycle, and the first one is called the lysogenic cycle. So HIV, for example, is a retrovirus. When it infects a host cell, which is actually a T helper cell as part of the host immune system, inside the T helper cell, the HIV virus actually makes double-stranded HIV DNA, and this double-stranded viral DNA can insert into a chromosome inside the nucleus of that host cell. So it doesn't necessarily cause immediate death of the host cell, because it's not a lytic cycle here. It's called a lysogenic cycle, whereby the viral DNA inserts inside the nucleus into an existing host chromosome, and the virus remains dormant for a certain period of time. However, the lysogenic cycle, whereby the viral DNA remains dormant in or as part of a host cell chromosome, it can then switch into the lytic life cycle. So the lytic life cycle is where the viral nucleic acid, for example, viral RNA, is then used and translated to make viral proteins. Now the viral proteins can then assemble into the new viral capsid, the protein coat that surrounds the nucleic acid. So these are called virions that are produced inside the host cell. So many thousands of virions are produced normally in the host cell. And quite often these new virus particles will emerge either through budding from the host cell membrane out of the host cell, or they might actually cause the host cell to lyse open and burst, which obviously would release the virus particles as well. So there's two main types of virus life cycle. The first one is called the lytic life cycle. Now in this example, we're going to talk about bacteriophage, and this is a type of virus that actually infects and replicates within bacterial cells. So here the host cell is actually a bacterial cell. So the virus inside it, its nucleic acid, will actually be here double-stranded viral DNA. So the bacteriophage has a capsid, and that binds to the surface of a bacterial cell. So the capsid proteins will actually recognize a certain maybe a receptor on the surface of the bacterial cell. Once the bacteriophage is bound to the host cell, it injects its double-stranded DNA here, and that's obviously into the cytoplasm of the bacterial cell. Remember, bacterial cells are prokaryotic. They have no organelles, they have no nucleus, no mitochondria. So here the double-stranded DNA enters the cytoplasm. Now actually this viral DNA does circularize, and then two things happen to the viral DNA. One is that the viral DNA is replicated. Now we can see here in green, replication of the viral DNA makes many new copies of the viral double-stranded DNA. And that's going to be important because obviously new virus particles, there must be lots and lots of the virus DNA to be encased or encapsulated within the capsid, the new capsids. Now the second process to occur is that the viral DNA is transcribed into viral messenger RNA. Now that's important because the messenger RNA can then go and attach to the host cell ribosomes. And we know at the host cell ribosome, translation occurs, and the viral RNA is then translated into viral proteins or polypeptide chains, which are the new capsid proteins predominantly. So the new viral DNA can then be encased within the new capsids that are formed, and we get thousands of new virus particles. Now these normally emerge from the host cell, if it's a bacterial cell, by lysis. So the bacterial cell is destroyed, thousands of new virus particles are then released, and they can go off and infect other similar bacterial cells. Now in this scenario, we've also got what's called a lysogenic life cycle. Now here the bacteriophage binds to the surface of the host cell, a bacterial cell. The double-stranded DNA is injected into the cytoplasm as we had earlier, but this time the phage DNA, or the viral DNA, actually becomes incorporated into the host genome. So the host genome is a circular chromosome free in the cytoplasm of the bacterial cell. You can see the phage DNA has inserted into that circular chromosome. If I just highlight this in green here, there you go. So the viral DNA remains dormant in this case. Now, if some sort of stressor occurs to these bacteria, and that might be, for example, a change in temperature or a change in pH, the viral DNA can be excised from the host chromosome. That means it can be removed once again. Now that viral DNA, it's double-stranded DNA, that allows the virus to enter the lytic life cycle. So we've got a change here from the lysogenic dormant life cycle to the lytic life cycle of the virus. Now the viral DNA, like we just saw earlier, it has to be replicated to make many copies of the viral DNA, because they will be covered or encapsulated within the new capsids that are being formed. And also the viral DNA has to be transcribed into viral mRNA, which is then translated at the host cell ribosomes, and we get the new virus proteins which form the capsid. So we get thousands of new virus particles again being formed from the lytic life cycle, and it results in lysis of the host bacterial cell to release all these new virus particles. So here, if you want to pause the video, you can maybe have a go at this activity, and this just shows the two different life cycles. Now again, here we've actually got double-stranded DNA within the virus particle. If the virus did have RNA as its nucleic acid, such as HIV, which we know infects T helper cells in humans, well, that RNA, if it's to enter the lysogenic life cycle, has to be converted first into single-stranded DNA, and then the single-stranded DNA is used as the template strand to generate the second complementary strand of DNA. So then we can see double-stranded DNA, and that can then integrate into the host chromosome during the lysogenic life cycle. So if the virus is a retrovirus with a single strand of RNA, the single strand of RNA has to be converted into double-stranded DNA for that virus DNA to then insert into a host cell chromosome inside the nucleus of the host cell. Now we can see here the host cell is actually a eukaryotic cell. It's got a nucleus this time. So here will be the nucleus of the host cell. So it's slightly different from the bacteriophage that we saw earlier. Now, obviously here we've got the virus attaching to the host cell membrane. So normally viruses will recognize a certain protein on the surface of the host cell. We've then got integration of the viral DNA. So this is viral double-stranded DNA in the cytoplasm that goes into the nucleus and it inserts into the host cell chromosome. So this is now called a provirus within the genome of the host cell. And this is the lysogenic life cycle of the virus. We know once the virus actually attaches and enters the host cell, obviously the nucleic acid has to be released into the cytoplasm first, and that's called uncoupling of the capsid and the nucleic acid. So you can see here the capsid's kind of released, and the nucleic acid is also free in the cytoplasm. So this is on the left-hand side, the lytic life cycle. We know the release of new virus particles, normally if it's the lytic life cycle, results in death of the host cell or destruction of the host cell. We know synthesis of new virus particles involves the host cell machinery, things like the ribosomes in the cytoplasm of the host cell, where translation of the viral messenger RNA does take place. And we know if the virus did enter the lysogenic life cycle, well, the DNA quite often is excised from the host cell chromosome, and that can then be transcribed into mRNA, and that can be used in translation and protein synthesis. Now, just one point of reference, if you look at the box top left, quite often in human infections, when a cell is infected with a virus, as the virus emerges from the host cell, it actually becomes covered in part of the cell membrane from which that virus emerges. So viruses like Ebola and influenza, when they emerge through what's called budding from the host cell membrane, they become encased within that cell membrane. And that's very important for the virus because it makes it less likely that the virus is recognized as foreign by the immune system of that individual. Because obviously if it's covered within cell membrane that's actually from originally the host cell, the human cell, it's less likely to be recognized by the different immune cells, the white blood cells within the blood circulation and the lymphatic system. Now it's important to understand that if a virus does enter the lysogenic life cycle, like human papillomavirus in the epithelial cells that line the cervix, or HIV in T helper cells in the blood and lymphatic system, when the viral DNA actually inserts into a host chromosome inside the host cell nucleus, if that viral DNA inserts within certain genes, it can lead to that host cell becoming cancerous. Now, cancer is defined as uncontrolled cell division. So there's two main types of genes that make a protein, a healthy protein, that's needed in the regulation of the cell cycle. Now these are called proto-oncogenes and tumor suppressor genes. So here we've got proto-oncogene and a tumor suppressor gene. Now the tumor suppressor genes, like the name suggests, the protein that's expressed from this gene, it helps to regulate the cell cycle. and it kind of puts a brake on cell division. So it helps to regulate or slow down cell division to the correct pace. Now, a proto-oncogene quite often makes a healthy protein that does promote cell division, but very, very highly regulated cell division. So imagine the viral DNA did insert either into, so we put here viral DNA, into the middle of a tumor suppressor gene. Well, that's going to change the sequence of codons in that gene, and it might actually change the reading frame of the gene. So a non-functional protein might then be expressed from this mutated gene because of insertion of the viral DNA. Now, if there is a mutant protein being formed that's non-functional, well, the protein, it may no longer be able to act as a brake on the cell division. So if there's no brake and no regulation of cell division, it can quite often mean that the cell goes into uncontrolled cell division. So if the virus enters a tumor suppressor gene in the middle, it changes the base sequence or the codon sequence, and that can make a non-functional protein. There's no brake and there's unregulated cell division. Now, a proto-oncogene, when that gets mutated, that can also give rise to a slightly mutated gene called an oncogene. So again, the viral DNA might insert into the proto-oncogene, changes the codon sequence, and we get a mutated form of protein being expressed, and that can quite often dysregulate the cell division or the cell cycle, so we get uncontrolled cell division, and again, that can lead to cancers. So the picture at the bottom is cervical cancer, and that's caused by a certain strain or type of HPV called human papillomavirus. Now, this does go into a lysogenic life cycle. Viral DNA does insert into a host cell chromosome. If it inserts into, for example, a tumor suppressor gene, it's going to disrupt the gene. No functional protein is made, and so the cells can become cancerous. So you can see here the cancer tissue is one in which you've got uncontrolled cell division. Now, if these cancer cells can enter the bloodstream and pass to different areas of the body, we can get what's called spreading of the cancer, and it metastasizes, and that can lead to secondary tumors or secondary cancers around the body, and that's when the patient is in need of severe treatment, okay, maybe chemotherapy, radiotherapy. Now, this slide shows an old exam question about HIV replication and life cycle. Now, we know HIV is actually a retrovirus, and that's because it has RNA inside its capsid as the nucleic acid. Now, HIV will infect, bind to the surface of T helper cells within the human body. So the capsid proteins on the outside of the virus can recognize certain receptors on the surface of a T helper cell, and the virus is then injected inside the cytoplasm of the T helper cell. Now, once inside the host cell, the capsid proteins kind of break down. It's called uncoupling, and that releases the viral RNA. Now, actually inside the capsid is another protein called reverse transcriptase. So HIV is actually a lysogenic virus, and it's going to make double-stranded DNA, viral DNA, from the single-stranded RNA. So we can see here in this picture, X represents reverse transcriptase. So the single-stranded viral RNA, if I just do this in orange, acts as a template strand. DNA nucleotides align next to the exposed bases, here in green. Reverse transcriptase is an enzyme that will then link together the DNA nucleotides into a new single strand, so single strand of viral DNA that's complementary to the original viral RNA. The reverse transcriptase enzyme has also got the ability to break down the original strand of RNA, so that gets degraded, and that leaves the single strand of DNA. Now, the single strand of DNA here, if I just draw this in here, that now acts as the template, and we get DNA nucleotides aligned next to the exposed complementary bases. The reverse transcriptase also has DNA polymerase activity, so that can now also generate this brand new strand DNA that's complementary to the original single strand of DNA. So we now end up with double-stranded viral DNA. So I'll quickly go through that again. RNA, viral RNA, is released into the cytoplasm of the host cell. Inside the capsid was an HIV enzyme called reverse transcriptase. So we can put this in, reverse transcriptase. And as the name suggests, this enzyme catalyzes the reverse of transcription. So mRNA is used as a template strand to make single-stranded DNA, which is the opposite of transcription. The reverse transcriptase breaks down the original RNA strand. It leaves the single-stranded DNA as a template. DNA nucleotides are lined next to the exposed bases, and reverse transcriptase now links together the DNA nucleotides by phosphodiester bonds. That forms the sugar phosphate backbone of the new strand of DNA. So we now have a double-stranded viral DNA molecule that enters the nucleus via a nuclear pore. Inside the nucleus, it will now integrate into host cell chromosome. And that integration is fairly random. There is actually another enzyme called integrase that is found inside the HIV virus. So you can see these black dots originally. One of them is called integrase, an enzyme, and the other one is the reverse transcriptase. So I'll do that in green for what we had earlier. So the integrase enzyme allows integration of the double-stranded DNA into the host cell chromosome at a certain position. So this is actually a lysogenic life cycle on the left-hand side. Now, on the right-hand side, we know HIV can also be lytic because it can destroy the T helper cells inside which it is present. Now here, the viral DNA in the host cell chromosome gets transcribed into viral messenger RNA. So we can see here viral messenger RNA. That's released via the nuclear pore into the cytoplasm. Also, the viral RNA, it doesn't have to be translated, and this RNA here will actually be the RNA that ends up inside, so inside the new virus particles. So some of the viral RNA that's made is going to be translated. So we can see here translated at the ribosomes, or it ends up inside the new virus particles as part of the new virus itself. So viral messenger RNA, when it attaches to ribosomes inside the host cell, we know the ribosomes are going to be ATS because it's a eukaryotic host cell. It's a human cell. Viral RNA is translated into viral proteins. These are predominantly capsid proteins. The new capsid is then formed. So we can see here, this is the new capsid, and it encloses the viral RNA that was transcribed from the viral DNA in the host cell chromosome. So this here on the right hand side is a new virus particle. We get millions of these inside the host cell as part of the lytic life cycle. The host cell will actually be destroyed in this case, and these millions of virus particles are then released to go on and infect other T helper cells inside the human body. So the actual question below says, use the diagram and your knowledge to describe how new HIV particles are actually being made. Well, the new particles are being made as part of this lytic life cycle, the replication process on the right hand side. So all you really need to talk about is the viral DNA being transcribed into mRNA. The mRNA can exit the nucleus via the nuclear pore, and two things happen. It's either used to build new virus particles in the cytoplasm, or it's translated at the ribosomes to make new virus particles that form the capsid proteins that then assemble to allow new viruses to be made. So this is actually the mark scheme you can see beneath. Viral DNA transcribed to make messenger RNA. That's viral messenger RNA. New viral RNA is made that will end up inside the new virus. Messenger viral RNA carries the genetic code for the amino acid sequence of the capsid proteins. So you need to reference the genetic code being a sequence of codons. Each codon specifies a certain amino acid within the new viral protein. Translation occurs at the ribosomes, so they're the host ribosomes, ATS ones. We know viral proteins are then assembled into the capsid, so you could put here capsid. Viral protein and viral RNA inside the capsid are assembled into new virions or new virus particles, which then get released through the lytic life cycle. So we're going to talk briefly about influenza virus, and in a similar manner to HIV, influenza is also a retrovirus, and that means that the nucleic acid inside the virus particle is RNA. Now, influenza virus actually has 8 different RNA molecules inside the capsid, and we know RNA is single-stranded nucleic acid. Now, each of these RNA molecules inside the influenza virus codes for a different protein. Now, the green layer that you can see here is the capsid. Now, we know the capsid is an envelope that surrounds the nucleic acid made of viral proteins. So the green layer on this picture is the capsid envelope that surrounds and protects the RNA inside. Excuse me. Now, 2 main types of capsid protein that emerge from the virus that allow it to form this antigenic type that's recognized by the immune system of the host. They're called the N and the H protein, and these are actually capsid proteins. So if we look at these 2 proteins, they look like spikes that emerge from the capsid, and the hemagglutinin in red that we can see in the picture. So I'll just highlight these red proteins here. They actually project obviously from the green capsid layer because they are capsid proteins. Now, the H protein is responsible for allowing the virus to bind to a receptor, a glycoprotein, on the cell membrane of the host cell, and it allows the virus to enter the host cell. Now, the N protein that you can also see as part of the capsid, it's a capsid protein that extends from the green capsid layer on the picture. So these N proteins here, they allow the virus to emerge from the host cell. It allows exit of the virus from the host cell. Now, as you can probably see from this picture, when the virus emerges from a host cell, it kind of buds out of the host cell. So the capsid, the green layer on the picture, it actually becomes enveloped or surrounded by this pink layer, and that is part of the host cell membrane. So the pink layer that you can see on the picture is actually phospholipid bilayer, because that's from the host cell membrane as the virus buds out and emerges from the host cell once it's been replicated. So the only 2 proteins of the virus capsid that pop out of the this kind of phospholipid bilayer are the N and the H proteins. So these are the 2 proteins that cause the virus to have a certain strain or antigenic type. So the immune system of the host will recognize or will respond to both the N and the H proteins, which are part of the capsid of the virus that pop out of the phospholipid bilayer that was originally from the host cell membrane. Now, if we actually look at a cross-section of the influenza virus, inside the capsid, we can actually see the 8 RNA molecules. So if we just quickly number all 8 RNA molecules, what you might be able to see is actually this yellow circle on the end of each RNA molecule. Now, we know RNA is single-stranded, and RNA stands for ribonucleic acid. Now, this circle, this kind of yellow circle, or I've just highlighted this in blue, is actually an enzyme called RNA polymerase, and it's a viral enzyme here, and it's actually attached to the single-stranded RNA. So what happens is when influenza binds the epithelial cell that lines the trachea or the bronchi, it infects the host cell and it's injected into the host cell. The capsid breaks down, and the RNA polymerase, the viral RNA polymerase, helps to make a complementary strand of RNA. So these 8 strands are called negative strands. And when inside the host cell, these are copied by RNA polymerase into a positive RNA strand, and it's actually that strand of single-stranded RNA that goes and attaches to ribosomes inside the host cell, and that's where we get translation of the viral RNA and therefore expression of viral proteins that will form the new capsid. Now you can also see, hopefully, that the N protein, okay, is this one in orange here, and that's actually an extension of the capsid. So the blue layer is the capsid that surrounds the nucleic acid, and this yellow protein is actually the one responsible for exit from the host cell. One of the other capsid proteins is the H protein. Now this is the one in purple here. Now you can see that's an extension again of the capsid, this layer of blue proteins here, and that is responsible for entry of the virus into the host cell. So this glycoprotein, this hemagglutinin, will actually bind to a glycoprotein on the surface of the epithelial cells. They're complementary in shape, and it allows entry of the virus into the host cell. Now the pink layer that you can see, like we just mentioned in the previous slide here, this is actually part of the host cell membrane. So when influenza viruses emerge from the host cell, they bud out of the host cell, and they take with it a portion of the host cell membrane. So obviously the immune system of that individual, it will still recognize hemagglutinin and the N protein, neuraminidase, as foreign, because these are not proteins that you find in the human body. So we can still mount an immune reaction, and an adaptive immune reaction against the influenza virus. However, the presence of the host cell membrane here makes it less antigenic. So the layer I've just labelled in orange there is the phospholipid bilayer of the host cell. It's part of the phospholipid bilayer of the host cell. And then we've got the 8 RNA molecules there that you can see inside the virus. They're called negative strands. We know RNA polymerase, which is the viral RNA polymerase, will make the complementary strand called a plus strand, and it's those that get translated into the new virus proteins inside the host cell. Now, RNA polymerase works by, inside the host cell, the single strands of RNA, so imagine that's a negative strand, it will have RNA nucleotides align next to the exposed bases. So they hydrogen bond to their complementary bases. RNA polymerase then joins together the RNA nucleotides by forming a phosphodiester bond, and that then forms the plus strand. That is then released and it attaches to a host cell ribosome. And we get translation, we get virus proteins, we get new capsids being formed that encase new RNA that's also been made inside the host cell. Now in the exam we are provided with quite a lot of information for each question, and we're expected to use that information. So this question here in the middle of the slide says, using the info provided, why might the flu be more common in winter compared to the summer months? Well, if we actually look at the information we're given, it does say that influenza virus is transmitted through aerosol transmission in water droplets that are sneezed out or coughed out of certain people infected with this pathogen. But it actually survives better when the air is dry. And it also survives when there's low ultraviolet light in the environment. So why would the virus be more common in the winter months compared to the summer months? Well, we know actually during the winter months, the air is actually drier compared to the summer months. So obviously a lot of the water's frozen. We also know there's less sun in the winter months, so there's actually lower ultraviolet light. So the influenza virus is more likely to survive and not be damaged as it's being transmitted from person to person in these water droplets in the atmosphere. So in winter, the air is drier, there's lower levels of UV radiation, better survival of the virus, so it can be transmitted through the aerosol droplets. Now, one of the reasons why UV light damages the influenza virus is because RNA is actually quite susceptible to UV radiation. So RNA, being a single-stranded nucleic acid, will actually start to degrade and break down when it's subjected to high levels of UV light. So how could you reduce the risk of influenza transmission? Well, regular and thorough washing of hands, using and discarding tissues for coughs and sneezes, which prevents or limits aerosol transmission. And also vaccines can be fairly effective. However, we do know that influenza, because it is a virus, it does actually have quite a high mutation rate, and antigenic drift can then occur. So new antigenic strains of virus can emerge each year, and that's why a certain vaccine is not necessarily going to be effective, because a new strain of virus might be present that has a different surface structure, slightly different surface structure, so it's seen as a different antigenic type. So most vaccines will actually now contain about 3 different viruses or fragments of the virus that are injected into the individual at the same time, and they're normally the most 3 prevalent strains of the virus that are present at that time. So it's important to be able to define what the term antigenic type actually means. So we define antigenic type as different individuals of the same pathogenic type of virus having different surface proteins or different capsid proteins, leading to the generation of different antibodies in the human body. So slightly different strains of influenza virus will have slightly different shaped capsids because of slightly different proteins that make up the capsid. Now we know inside the human body, B cells are white blood cells or lymphocytes that have antibodies on their surface, and each B cell will have a slightly different type of antibody that binds to a specific, put here, specific antigen. So 2 different strains of influenza virus will cause 2 different B cell responses inside the human body. So we know there's many different antigenic types of influenza, and they emerge by 2 different mechanisms, the first of which is called antigenic drift. Now, antigenic drift, as the name kind of suggests, refers to a gradual change in the influenza virus that happens fairly slowly over time because of mutations in the viral RNA. So the reason we get mutations in viral RNA, such as the influenza RNA, is because, imagine this was the molecule of RNA, or one of them, called a negative strand. So we could actually put here some bases: AUC, GGC, CCA, UU, etc. Now, when this RNA strand is copied inside the host cell to make a new strand of RNA, we know the nucleotides align next to the exposed complementary base. So U, A, G, C. I'm going to put a G in here for the time being. G, 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 U, A, A. So these are complementary RNA nucleotides. Now, RNA polymerase, the viral RNA polymerase, links together these nucleotides into a new strand of RNA, complementary strand, and it's this plus strand that will then go off and attach to the ribosomes for translation. Now, if you look back at what we've just drawn, you can actually see that the G at position 1, 2, 3, 4, 5, so this position here, has actually been copied, or complementary base is put down as a G. So that's actually incorrect, because it really should be a C in this new strand of RNA that's being made against the original template strand. So that the sequence of the RNA plus strand should be UAG, CC, not a G, GG, GUAA. Now, RNA polymerase has no proof reading activity. That means if any mistakes are made in the new strand of RNA that's being made against the template strand, it's not corrected. So this actually here, this G, is a mutation. It's the wrong base that's been put into, or the wrong nucleotide that's been put into the new copied RNA strand. So that's called antigenic drift. Now this new strand of RNA with a mutation in, that might code for a slightly different polypeptide chain. The codon might be different at that position. And so obviously that might be a different capsid protein with a slightly altered shape. So it's important to understand that the two main types of capsid protein that get altered by mutation that actually affect how a virus enters or exits a host cell and therefore affects its antigenic type are the H and the N proteins that we mentioned earlier. So the virus surface protein called hemagglutinin, or the H protein, we know allows entry into the host cell, and neuraminidase N protein allows exit of the virus from the host cell. So when these viral RNAs, because we know influenza is made of single-stranded RNA, nucleic acid, if they get mutated because the viral RNA polymerase does not have any proofreading activity, it doesn't check the new strand of RNA once it's been copied, we know mutations can occur. These might code for slightly different versions of the polypeptide chains that get incorporated into the capsid of the virus, and so new slightly different strains of virus can emerge. So just to reiterate about how the mutations actually occur, we know new viral RNA is made against the template strand of RNA. Some mutations are introduced where the wrong nucleotide is inserted into the new strand. There's no proofreading activity of viral RNA polymerase, so the mutations quite often actually get expressed into a new version of the protein, and we get a different antigenic type of virus that emerges. The second mechanism of how new antigenic types of virus can emerge quite suddenly is called antigenic shift. Now, as the name suggests, this is quite a sudden shift or sudden production of a new strain of influenza virus. And it is more rare than antigenic drift, so the emergence of new strains of virus through mutation alone. Now, antigenic shift occurs when a host cell, epithelial cell, gets infected with two different types of influenza virus at the same time. So imagine we had, for example, avian flu virus. So avian just means bird. So these bird influenza viruses, like H3N8. And at the bottom, we've also got human influenza virus. So inside each of the influenza viruses, we know there's 8 RNA molecules. Now, one of these 8 RNA molecules will actually code for the H protein, hemagglutinin, on the capsid of the virus. So in the avian influenza virus, we can see the RNA molecule is drawn in orange here, and that's the genetic code that expresses or codes for the H protein. So here it's H3, that's the version of the H protein. So on the surface of the capsid, we're going to draw these capsid proteins, H3 protein in orange, because they're coded for by the H3 RNA inside the virus. Now we can also see that there's an RNA that codes for the N protein. Now we know the N protein is needed for virus exit from the cell. So here the N protein is called N8. So this green RNA, the N8 RNA, codes for the N8 protein as part of the capsid to allow the virus to get out of the host cell. So we can actually color code the capsid proteins corresponding to the type of RNA inside the virus. Now at the bottom we've got the human influenza virus. Now we know again there's 8 RNA molecules. One of them codes for the H protein, but in this case it's actually H2, it's not H3, H2. So the capsid protein, the H protein, hemagglutinin on the surface of the capsid, we can actually color in blue because that is coded for by this specific type of H2 RNA inside the virus. Now we can also see there is an RNA, one of the 8, that codes for the N protein, but this RNA codes for the N2 capsid protein. So the purple strand of RNA, the N2 RNA, is the genetic code for the N2 protein on the surface of the capsid. So we can color code this in purple. Now imagine we had, for example, a pig as the host. So the pig might get infected with the avian influenza virus H3N8, and it also might get infected with the human influenza virus H2N2. So imagine one of the epithelial cells that lines the trachea or the bronchi or the bronchioles of the pig simultaneously had infection with both this bird influenza virus and the human influenza virus. So both viruses attach to the cell membrane, they get injected into the host cell, and then what we'd have is not 8 different RNAs, but we'd actually have 16 different types of RNA inside the host cell, 8 of them from the bird flu virus and 8 of them from the human flu virus. Now, when new virus particles are formed inside this host cell in the pig, we know each virus particle that is formed will have 8 RNAs within the capsid. However, there could be a mixing of the RNA, so it has to have all 8 RNAs, but it only has to have one version of the H RNA. So imagine, for example, when a new virus particle was formed, the RNA that codes for the H protein was the H3 RNA. So you can see here that's the orange strand of RNA that originated from the bird virus. So that would code for the H protein as part of the capsid, which is the H3 protein. So here it's in orange. We just color code them. And also one of the 8 RNAs inside this new virus is obviously going to code for the N protein. Now here it's the purple N protein N2, and that originated from the human influenza virus. So there's been a mixing of RNA inside the host epithelial cell of the pig. So the N2 RNA would code for the N2 protein as part of the capsid. So again, we'll color code this. So we can now see this new virus has emerged, which has a combination of the RNAs from the bird and the human influenza viruses. This is a brand new type of influenza strain of virus. We can put, it's actually a new antigenic type. So new antigenic type. Now this has a new structure to its capsid. So some of the proteins will be from the bird influenza virus, and some of the protein is from the human influenza virus. Now, most people will not have immunity to this new strain of virus that's emerged very rapidly, because obviously they will have memory B cells to antigens that they've actually encountered before. However, they won't have encountered this virus before because it's a brand new strain of virus. Its capsid structure is different from both the bird influenza virus and the original human influenza virus. So because people don't have ready-made immunity to this new strain of virus, because it's a new antigenic type, they might actually get quite sick with flu. Now, older people and immunocompromised people and younger children might be more affected by this new strain of virus. And so if it is, or it does lead to an epidemic, if the pathogen spreads across international boundaries, it can actually result in what's called a pandemic. And that's where we get lots of people who fall very sick very quickly with this type of flu.